It is so exciting to be here today. My career started in the society, and I'm so grateful to all of the organizers, Shenner, Steven, Stefan, and Martin, and of course, Rudy and Gerhard. Thank you for giving me the chance to tell you about some of the work that we've been doing. So in some ways, I feel very sheepish standing in front of you to talk about this topic because honestly, I'm a neophyte. We started work in this area maybe 7, 8 years ago. On the other hand, why not take the opportunity to tell you about some of the most fun that I've had over those years? I want to start out by emphasizing how widely vast biological systems are. So I start with the tree of life. This is a depiction of all of the species that have existed. Most of them are in fact extinct. And the kinds of things that I work on are a tiny speck and a tiny speck over here. There are so many different problems that members of our community have been working on in this area that I will not even remotely touch upon. I won't talk about DNA sequencing. I won't talk about DNA storage. I will talk very loudly, apparently. I won't talk about timing channels or directed information for neuroscience, et cetera, et cetera. There are a huge number of problems that we could be getting involved in. So I encourage you to think about them. I recently came across this quote, and it seemed absolutely perfect. So if you'll indulge me, I'm going to read it to you. Will it ever happen that mathematicians will know enough about the physiology of the brain and neurophysiologists enough of mathematical discovery for efficient cooperation to be possible? And this quote is from none other than Jacques Hadamard. I personally used Walsh-Hadamard codes when I was studying multi-user CDMA. I've looked at the Hadamard inequality on determinants of matrices when we were looking at optimizing space-time codes. He's made tremendous contributions. As you can see from his birth and death, this was a man who had his foot in both the 19th century and the 20th century, and he was also a bit of a philosopher. I'd like to put forth not an original idea, but that which was given by Toby Berger during his 2002 Shannon lecture, where he said, you know, at the birth of information theory, everybody wanted to adopt it as a methodology to use to explain what was going on in their field, and Shannon was very dismissive of this. And Toby put forth the following hypothesis that maybe that was true because we were not ready yet, because we did not have the measurement or observation tools that we have now that enable us to have data sets that allow us as communication and information theorists to use our modeling and strategies and abstraction and even potentially optimization. So I'm going to stand on the shoulders of giants and say, yes, I think we're ready. So many of you are very familiar, perhaps, with Shannon's master's thesis, which put forth the capabilities of Boolean logic in terms of computation. It was groundbreaking work. Some people say it helped start fault tolerance in the area of computer engineering. But I wonder how many of you have actually looked at Shannon's PhD thesis. So being a professor, I feel it's important for me to give you homework. So homework number 1 is read Shannon's PhD thesis. And he actually tried to create an algebra for theoretical genetics. It's basically combinatorics. He was looking at how genes get passed down given an arbitrary initial frequency. And to be truthful, if he had a handle on modern matrix theory, this would be a much shorter thesis than it was. He was not happy with the work. It never got published. But we can at least say that Shannon, too, thought about how his tools and techniques could be used to influence disciplines outside of communications and engineering. I like this particular picture. It's with Claude Shannon and his wife. It's at Brighton, which was where my colleague Saul Gonol gave his Shannon lecture. I'm sorry, I'm going to get a little emotional. We lost Saul last year, and interestingly enough, or coincidentally, it was the day after the centennial of Shannon's birth. And I had interviewed him a few days before that about Shannon, so it was kind of an eerie situation there. And this is also my colleague Bob Schultz. As a plug for something that I think we should be thinking about, both Saul and Bob worked on comma-free codes, and I want to put this forth out as a second homework problem of something we could think about in terms of these biological systems, because these are codes that self-synchronize, and I think the question of synchronization is a very important one in these biological systems. Okay, so why you might want to study biological systems if you are an information theorist or a communication theorist. A lot of these are kind of obvious, perhaps. Maybe not taking ideas from Mother Nature and seeing how we can improve man-made systems. But one of the new areas that I've learned about in working with my biophysics colleagues is that when they do experimentation, it's extremely expensive. And when I say expensive, I mean money, dollars, but I also mean time. I tell my grad students, be happy you're not an experimental biologist. You don't have to spend 72 hours in the lab turning knobs and making measurements and making sure everything works out. You can spend 72 hours in your pajamas proving theorems at home, which is a much more comfortable thing to do. So the point is, and my colleagues are very interested in this, if we can somehow point to what the interesting regimes are so that they can have shorter experiment times or look at only important ranges of environmental conditions, this will have a huge impact on their research. And so I suspect that this is one of the reasons why that they talk to us and try to educate us about the problems that they're interested in. So these are some of the big questions that we are attempting to ask and answer, if you will. And truth be told, I think at the end of this, I have far more questions than I have answers. But I definitely do have optimism and hope that the tools that we can bring to bear on these problems will have an impact. It may not be today, it may not be in 5 years, but in 20, 25 years, we can do something that will be potentially profound in collaboration with these kinds of individuals. So, are our tools powerful enough to say something interesting? Can we come up with the right simplifications and abstractions so that we can use our tools? Are we adept enough to understand how to translate biological constraints into engineering constraints and use them in order to design our systems? And my favorite question is, are there regimes in which we can say Mother Nature is in fact optimal? And I'll tell you that my colleague Mo El-Najjar always tells me, Mother Nature is not optimal, she just does what works. But we will see. So this is the talk. If you remember nothing else, you can leave now. This is what we're going to study. And we're going to study point-to-point systems, multi-hop cables, and then more interesting multi-terminal networks. Again, I put forth to you that these are structures that you are all familiar with and may even have some exciting results on, which could be brought to bear in this space. For me, this is exciting because when I started out, or when I finished my PhD in 1994, it was legitimate to study point-to-point communications. Nowadays, when I talk to my students and we set out problems, we realize that we have to look at networked physical layer, that point-to-point is no longer legitimate. A lot of it has been solved. But in biological systems, there's still a lot to do. So a question we should ask ourselves is, in this context of whatever problem it is that UBLI wants to study, what does it mean to say communications? What do we want to study? And so my second piece of reading homework is one of my favorite papers by one of our own, Chris Rose and Sarah Mian. This is a lovely paper which essentially lists, I would say, hundreds of potential research problems for all of you to work on. It looks at how communication theory comes into play in multicellular biology, and as just a simple example of how we can transmit information, here's a cartoon of a mitotic spindle, and what's put forth in this paper is that the angle of this particular spindle conveys information. As it rotates around, we can convey information. And so this is not something that potentially many of us may have thought of, that some physical property like location or presence is actually informative. So I told you at the beginning, biology is vast. Even for the problems that we've looked at, there's tremendous diversity. So what I have here are examples of signaling molecules, if you will, or signaling quanta that we have studied in our research. We've looked at the transport of electrons. We've looked at the impact of glucose on the production of ATP, and I'll talk about this and more. And when we talk about quorum sensing towards the end of the talk, I'm interested in this very tiny black speck here, which is the LASER protein, which is the autoinducer that induces quorum sensing. I don't expect you to understand what any of this means, but just to recognize that even pictorially, these things are very different objects with very different properties. And as a result, the models that we develop have to take these distinctions into consideration. If I look at how cells interact with each other, we see there's also a wide variety of diversity. They can exchange molecules by being next to each other. If there's further distance from each other, they can actually diffuse molecules into the environment that can be sensed by another cell. Or they can do something much more active, including directly transferring genes from one cell to another. And we do see this in bacterial populations. What's exciting about this is that depending on the signaling interaction that the cells undertake, there are potentially very different consequences. Can the cells now cooperate to form a sensing mechanism? Can they actually migrate, which is what happens with these very simple organisms called slime molds. They actually physically move as a function of the signals that they send, and they can fundamentally change their behavior, morphology, phenotype, et cetera. So biology is vast, but the good news is that the tools that we can use are also vast. So I maintain that the strategies that I can use as a communications engineer, my toolbox has just gotten much larger due to the different things that biological systems can do. So let's start point to point. I won't do justice to all of the people that I have worked with on this, but for point-to-point communications, I'm indebted to my two Sharif collaborators, Masume Nasiri Kanari and Amin Gohari. And so we decided to focus on molecular communication in particular. This, in a way, is one of the most engineering types of problems you can address. But here, too, there are choices and diversity. I can diffuse molecules into the environment. I can do something much more engineered. This is a lab on a chip that was devised by new friends at Johannes Kepler University in Linz. I visited them in the spring. Vernal Hassmeyer and Andreas Springer are interested in doing computation using droplets. And so they communicate and compute by putting droplets through channels. So this is a microfluidic system. You can also do something even crazier, which is to use molecular motors. So this is a depiction of a molecular motor. If you look, the molecule is much bigger than the motor, but it's walking along a protein strand and actually transferring a molecule from one end to another. And there are different groups that are working on this kind of thing as well. So let's say I want to build a molecular communication system. So I'm focusing even more now. I want to focus on diffusion-based communication, that first cartoon that I showed you. You can argue that it doesn't require infrastructure like one of the flows or the walks. And it has some properties that are very similar to things that I'm used to worrying about in wireless communications, for example, intersymbol interference. And in the hopes that maybe we could design one of these systems that could be built in, say, 5, 10, or 15 years, I want to focus on strategies that have very simple complexity, all right? So I don't want to do things that are too complicated. Now, the first thing that you should ask, or the first thing I always ask, is if I'm going to build a transceiver system, what's the channel? What am I designing to? And that is the first thing that we have to ask. Again, more diversity. Depending on how I want to send my molecules, I can vary concentrations, I can change the molecule type, or I can even change the timing. But as I said, I have intersymbol interference. And so these molecules persist in the environment for an incredibly long time. So I might want to send a high concentration, but it might continue to leak into the next symbol, if you will, and my low concentration might look like a high. Even though we might think that molecules could potentially be orthogonal to each other, they can interact. And so even two molecule types means that there are strategies I have to design at the receiver in order to ensure that I'm receiving my zeros and ones properly. And timing, I think it's not too hard to argue that this will complicate things dramatically. Okay, so I want to walk through a model. I did not design it, but I use it, and I like it quite a lot. So let's go to basics. Let's assume that I have a vessel. It contains molecules. And I have some capability of gating things at the front of this vessel so that molecules can be released into the environment. I'm going to assume that I have Brownian motion, that my molecules don't interact or change with each other. I have an infinite medium, and the only thing that my molecules can possibly run into is the receiver. Okay? And information is going to be encoded in the release time and eventually in concentrations. Okay? So you can show that the noise in the release time can be modeled as this so-called inverse Gaussian. And here is an example of what different PDFs look like. This is derived from things that we all studied in graduate random processes, and that is when we looked at Wiener motion, we modeled it as a Wiener process with independent Gaussian increments, and the statistics of that Gaussian of the independent increments are defined by the environment, molecule type, fluid, velocity, et cetera. And again, there are those in our community that have truly developed this model and used it to try to do capacity computations. There are a number of things that we can say. So now I'm in a regime where things look very familiar to what we're used to in communications, where I have my release time, I have my receive signal with this noise that takes on this inverse Gaussian probability density function. And I'm actually going to simplify things a little bit more and not work with the AIGN. As Stefan Moser can attest to, this is a messy density to work with. So I am going to be less ambitious. What I'm going to recognize is that I can use this AIGN distribution in order to compute the probability that a molecule is going to hit a receiver. And in doing that, I can change the shape of my problem into something that's more tractable. So I am going to abuse notation a lot in this talk, and I apologize. So now let's assume that x is the rate of molecules exiting my storage. So I have a sequence of rates, okay? And the number of molecules that get dispersed is a Poisson random variable with those rates, okay? And so here's my information sequence. Now my received signal is also a Poisson random variable with the following rate. And if I were in a smaller venue, I would ask someone to raise their hand and tell me what operation is going on inside these braces, but it's early, so I'll spare you this. This is something we've all seen, that effective rate at the receiver is simply the convolution of that sequence of hitting probabilities that I know how to compute and my information sequence. And so now I'm in comfortable communication block diagram universe, right? Where my information sequence goes through an LTI channel followed by a memoryless Poisson system. And this is something that Masume and her students came up with. Now I can do things as a communications engineer. So let me do a little bit with this, okay? So again, I have my communications engineer hat on. I can do things that are similar to what I've done for RF. That is, I can look at a system where I have an input stream that governs the rates of the molecules exiting the storage, and maybe I will have a simple counter at my receiver, and then I can put a little bit of computational complexity at the receiver, and maybe what I will do is have an adaptive threshold, right? So if I'm doing binary communication, maybe I'm sending nothing for a 0, I'm sending something for a 1, and since I know something about the intersymbol interference and my hitting probabilities, I can use this to decide at any given time what would be a good threshold for me to use to disambiguate between a 0 and a 1. And so this is something we can do, and we can say some nice properties about this kind of strategy. Let's do something a little more interesting. So one of the things that, again, in retrospect is obvious, but I will state it to you, I don't have negative molecules. My molecules don't really have phase per se, all right? So if I want to design something like a precoder, I can't actually invert a matrix and use that to design signals to send through my, my vessel, right? I can only do things with positive objects. So something I can do is to modify what I do at the transmitter. I assume that I know what my sequence of rates is going to be, and if I'm doing, say, 0s and 1s, binary communication, I can actually pre-code by essentially water filling on the interference that I'm going to experience at the receiver. And then I can have an extremely simple detector at the other side, which is a constant threshold to decide between a 0 and a 1. And we can actually show that this water filling strategy is optimal under certain kinds of conditions. So you might ask, okay, how well does all of this work? And so I can look at different strategies. So what I have here are two schemes that ignore, first one that ignores the interference and has no memory. It has no memory but guesses about the interference. I have that adaptive transmitter strategy that I showed you here, and then the adaptive receiver strategy here. The 2 points that I want you to take away from these pictures is that these two pairs of lines are close to each other. The top line always corresponds to 2 bits of memory, and the one below it to an infinite amount of memory, which means that 2 bits is more or less enough for me to design a system that operates near optimally. The other point you should take away is that my adaptive transmitter does not work as well as my adaptive receiver. However, part of this is a complexity performance trade-off. The other thing that I want you to notice, if you haven't, is that we are looking at very low data rates. We're not going to be transmitting video in vivo, okay? We're going to be using these kinds of schemes for telemetry, maybe signaling for other kinds of mechanisms. And so, again, another question you could ask is, Ubli, how science fiction is this? And I maintain it isn't. So this is work that was published in 2012 out of the Wyss Institute in Cambridge, Massachusetts. And this is a vessel that's a DNA cage. What you see inside are molecules, and under the right environmental conditions, this cage will open up and release the molecules. And I maintain if we can do this now, then my simple transmitter-receiver system is definitely doable within 5, 8, 10 years. So we're not as far into the science fiction realm as you might think. So, of course, it's, we're at ISIT. You say, look, you've got this LTI Poisson channel model. Can you now compute capacity? Can you do something? And I'll say we can do something maybe, all right? So we, as many of you can attest to, computing the capacity even of a simple Poisson channel is quite challenging. But we realized that by looking at the symmetrized Kullback-Leibler divergence, we could come up with some simple upper bounds. And we conjecture that these bounds are tight at low SNR. And so just to give you, evidence that things are simple, for a simple memoryless Poisson system, this is the upper bound you can compute. Of course, you will want to optimize this over the input distributions, but the key thing is that this is a simple expression, all right? So this is my proof that the bound is simpler than what has been done before. This is still open, okay? So homework problem number 2 is to try to come up with a better bound. Okay, I told you at the beginning that I was not going to allow molecules to have anything happen to them, to disappear, vanish, or interact. But again, in my enlarged toolbox of what I can do in a biological system, I can use enzymes. And so enzymes collect substrates from the environment, create new products, and these products themselves can change behavior. So for example, enzymes can fundamentally change reaction rates. They can speed them up or they can slow them down. And so individuals have been looking at how you might use enzymes in molecular communication systems in order to deal with intersymbol interference. I don't have an enzyme for my wireless communication system, but now, as I said, I can do a lot of different things. Others have looked at higher layer problems in terms of networks. So this is one more strategy we can use. Something that I completely glossed over, but I alluded to at the beginning, is the issue of synchronization. Everything that I talked about before in terms of my transmitter and receiver assumes that I know when symbols start and I know when they end. This is going to be a profoundly challenging problem in these molecular communication environments. There are those who have taken a classical engineering approach and have tried to estimate delays and synchronize that way. But I maintain that we should look at what Mother Nature does. So this, unfortunately, is not very well shown, but these are fireflies, and as we know, in summer nights in neighborhoods where these occur, eventually their firing or lighting up synchronizes together. And this is essentially a sort of collective phase lock loop where they're making minor adjustments to their oscillators. And people have studied this in the context of sensor networks and also in the context of its biological systems. What I have here is a plot from a paper from Elisa Franco. She's at UC Riverside. She is a control theorist by training and has essentially become a synthetic biologist. And they have actually built biological oscillators that do synchronize with each other. And this is an example of some of the results that they have. So again, we do have the capability to do some of these engineering things that we're used to, like synchronize, but they're going to be done in a fundamentally different way, which means we have to design our systems in a different way as well. So to motivate the bacterial work that I've done, I wanted to point to this new image of the tree of life. It's new and improved for reasons that are important to our biological friends, but it's important to me for the following reason, and that's because of what's in the triangle here, which corresponds to bacterial life, these unicellular organisms. And so the key thing you should see is that they outnumber us many to one. And in fact, in terms of aggregate mass, there's more of them than there is of any other living thing on Earth. Despite being unicellular, we don't really understand them. But because they're unicellular, they are actually relatively easy for my biophysics colleagues to manipulate and therefore create strains that allow us to study new and interesting principles. And for me in particular, because of my biophysics colleagues, I'm interested in the role that they play in infection and also microbial fuel cells. So I want to tell you a story about a phenomenon which was intriguing. This is Philip Meisman. He is a faculty member at the Vrije University in Brussels. What he has in his hands is a tube of sludge, sediment, collected from the ocean floor. And what's in that tube is something that they think looks like this. This is a collection of bacteria, end to end, holding hands, if you will. And in the process of creating this cable, they've created a sheath around themselves that allows them to transport electrons between them very efficiently. The biophysicists, the microbiologists, they do not understand why this happens. It's an unknown. And the reason why it's unpredictable to them is the amount of nutrients at the top of the cable is fundamentally different than the amount of nutrients at the bottom. So somehow, for some reason, these bacteria are incentivized to hold hands and be in this vertical environment, even though the amount of food at the top is different than the amount of food at the bottom. Now, I maintain that bugs don't do anything except to be happy, all right? And so the question is, what about this structure promotes biological fitness, okay? Now, if you look at this, what do you see? A multi-hopped communication channel. At least that's what I saw. And we've been worrying about multi-hop communication channels for quite some time. I want to point out to two individuals in particular, because they're here today. Wenyi Zhang was a postdoc in my group, and upon reflection, I realized he had done a ton of multi-hopped work while we were working together. And Nicola Micheluzzi is the one who really put this bacterial work forward. He made it happen. He was also a postdoc in my group. We rely heavily on our biophysics colleagues. Mo El Najjar is a biophysicist, and Sahand is his former student, now postdoc. Okay, so let's go back to this picture. I have this multi-hop communication network. I have electron donors interacting with electron acceptors, and these cables are multiple centimeters long, which means we're talking about thousands of different bacterium holding hands together. Why do they do this? Why is it this length and not 1 millimeter or a meter? Why do these things happen? And so the question is, can our communication and information theories help us answer these questions? Okay, so reading assignment number 2. This is Charlie Desor. He was a faculty member at Berkeley. I learned linear systems from him when I was doing my master's degree. He was also my father's master's advisor and had a profound impact on how my dad teaches and does research. This, I think, is an unappreciated or underappreciated piece of work. In Charlie's PhD thesis, he studied what they called cascaded channels. It's a beautiful document. It talks about the impact of delay. It presages this notion that the capacity of a multi-hop link with infinite block length corresponds to the worst case link's capacity. He talks about the impact of processing at the different relays. I would encourage you to read this. So let's think about Charlie as we move forward. So one thing that I have not mentioned, and I apologize because I was trying to figure out how to put all of these things together, is that these biological systems don't just communicate. We can use communication as a proxy for many things, but they don't just communicate. They also have to sense. And so putting my engineering hat back on, I have to recognize that there's a tension between sensing and communication. So let's look at some baby problems that we're used to seeing. When we think about communication and we look at the classical result, we will often model our channels as something that we know, all right? And so what we want to then do is put as much randomness as we can into the input in order to maximize the capacity of the channel. On the flip side, I was boasting that I was going to have much deeper technical difficulties than Andreas, so this still might happen. On the flip side, if we do estimation, we do something that's exactly the opposite. If I want to learn the channel, I send a deterministic signal across it in order to learn it. Okay, so on the one hand, if I want to communicate, I want random. If I want to estimate, I want deterministic. So these two things are clearly in conflict with each other. So let's remember this because I believe these systems both sense and communicate. The other question has to do with relay processing, all right? So what are the different things I can do at my relay as I'm communicating across this multi-hop case? And there are different strategies I can use. I can scale my received signal, amplify and forward. I can do what I'm going to call reconstruct and forward, because both in the communication case I can decode, and then in the estimation case I can estimate. So why am I raising these processings? The point is that when you sense, everything changes. So when we've looked at communication problems, we've worried about things like how do you schedule packets to manage interference, if you are trying to balance between your rate, error exponents, and delay, what's the optimal number of relays that we should have? And again, this problem is going to raise its head again when I worry about what the optimal length of a bacterial cable is. And we've also looked at sensing problems. So this at the time, what we did in the heyday of sensor networks, and it seemed kind of like a toy problem, but I'm now realizing that it has impact on what I want to do today. Assume we have this multi-hop link, and I have a series of channel states that I want to estimate. And I want every node to estimate all of the preceding channels before it, okay? I can look at the asymptotic mean squared error and ask myself, how does it decay as a function of the SNR? And this exponent tells me how good or how bad my system is. What we can show is that for estimate and forward, we get a decay rate or an exponent that is absolutely less than 1, but if I do amplify and forward, I get an exponent that equals 1. Why is this worthwhile mentioning? Well, this is the opposite of what happens in communication, right? In communication, decode and forward is the thing that I want to do. It's what's going to achieve my capacity, and amplify and forward is suboptimal. So we have this opposite relationship in the multi-hop case between sensing only and communication only. But of course, this is not the whole story, because in both of my cases, I don't take into consideration the fact that there's coupling between the links, and this is something I have to worry about. So again, sensing matters, and I want to put forth an advertisement for communication over channels with state, a classic problem whose time has come back again, but now with the issue of sensing there. So before I can talk about communication with, over channels with state, I want to come back to my problem, okay? So let's never forget, I want to understand why this bacterial cable exists and why it's as long as it's supposed to be. So one of the things that's interesting about these biological problems is that, in a way, capacity is not the bottleneck, but, and I hate to say this because it sounds kind of holistic and very Los Angeles, but happiness or wellness is the thing that I need to worry about. And ensuring that my bacterial cable is happy is actually going to change how much information, if you will, I can push through this cable. So we had many interesting conversations about what it means for a bacterium to die. Do they really die? Are they not dead? We talked about the undead. But before I can talk about my cable, I need to understand what happens within a single bug. So remember I told you about this notion of transporting electrons and electron transport and that sheath that's formed around the bacterial cable so they can transport electrons. Again, you know, a biologist tells you this and you say, oh, that sounds interesting, but I don't know what it means. But then they say, look, we know that within a single cell, we have to transport electrons. We need a proton gradient. That proton gradient is what's going to create the energy currency of a cell, ATP. And this has been well understood within a single cell for many years. The thing that is novel here is the fact that now, in these bacterial cables, they are transmitting electrons between each other. And this is something that people did not realize could happen. So what do I want to do? I'm going to put my communications engineer hat on. I'm going to not do what has been done in the past. We are not the first to put forth the idea of mathematically modeling biological systems. They have gone to first principles where they talk about diffusion, differential equations, et cetera. What ends up happening is you have a very long list of differential equations, you plug them into a huge solver, and it becomes almost like experimentation. You turn knobs for different parameter values and you see what happens. But I want to optimize things. I want to get things abstract enough so that I can prove something and see what my optimal value should be. So I'm going to look at both chemical and electron concentrations. They're going to be measured as Qs. And then I'm going to take all of the things that I can't model well and make them stochastic. Okay, so this one slide is the result of 18 months of conversation. And there was much arm wrestling. I have a few scars, but I won't show them. You know, can we take this idea away? Do we have to model that? No, we can. Yes, you can. Back and forth, back and forth. And so what we have is this model for a single bug. It has 4 Qs. The one at the beginning and the one at the end is electrons coming in and electrons going out. We also have some leakage. It's the 2 in the middle that I want you to focus on. There's 1 Q that corresponds to NADH production, and this is the process that takes molecules from the environment and converts it into electrons, if you will. And then I have this other pool, which is my ATP production pool. This is the important pool. If this Q level goes to 0, the bug dies, bad. And if it gets too high, it's actually also a little bad, because what happens is that the bacterium slows down in its production of ATP. Bugs do not overeat, okay? I wish I could say the same. But it is something that I have to model. The other thing that's hidden in here as well is the fact that there are all sorts of equilibria that we have to maintain. So when I come up with my queuing equations, I have to make sure that certain inputs and outputs are equal to each other. The point is that I have a list of biological constraints that I need to take into consideration, okay? So we wanted to validate this. I knew this was going to happen. See, Andreas, don't worry. Someone will have more troubles with electronic stuff. So one of the things that we wanted to do is validate this model. Okay, let me see if I can do this more quickly. And in order to do that, I'm sorry, I'm just going to flip through. I hope I don't cause any dizziness. And one of the challenges in validating the model was actually getting experiments that reflected this cable that we had looked at. So people are very interested in it, but it's hard to do. If my colleagues ever create the measurements that give me NADH and ATP values simultaneously, I will have the least important author placement in a Nature or Science article, but we're not there yet. What we were able to do is to take data from the literature, where we have glucose here, and it's measuring the levels of ATP. And what you can see here is that our model matches the experimental data extremely well. In fact, our biophysicist did not believe Niccolò when he put the data forward and said, look what I've been able to do. This is a simple system with only 2 Qs, but we have very good match. Okay, but I don't care about one bug. I want to look at many bugs holding hands, right? And so the question is, how do I deal with this? And this is where we put our information theory abstraction hat on. And we say, well, let's consider the entire cable as a channel that has some channel state. And can I look at information as a proxy, or capacity as a proxy, for how many electrons can be pushed through the system? Okay? And so I'm going to have a signal also where I'm going to modulate the rates. It's going to create a Poisson random variable. It's going to be sent over this channel. And I want to see what I can do at the receive side. This is definitely an abstraction. I also have to take that not overeating into consideration. So I have a function A of S that we call the clogging function that is going to look at what the channel state is, and if it's too high, it's going to give me a low value, and if it's low, it's going to have a high value. And this is going to impact what I'm going to do. And so the key thing here that I want to see, so we do a lot of approximations, is that my input process is now this Poisson process where the state of the channel affects what I can send. So I want to send this lambda of t, but the cable is going to enforce this A of S on top of me, all right? And so if this is the case, how well can we do? So there are a lot of technical statements that you can make, all right? These technical statements are important in the sense that they allow us to simplify our optimization dramatically, so we can compute the rate that we want. The one I want you to focus on is this one at the bottom. So again, engineering hat on again. You end up looking at a dynamic program in order to compute the capacity. And so it's legitimate to ask, well, what happens if I do the engineering version, which is at one step ahead computation of rate versus a many step ahead over the entire horizon? And what we can prove is that the average rate of this myopic scheme is actually greater than or equal to the average rate for the optimal scheme. Now, at first you should say, wait, wouldn't that imply that the achievable rate of this greedy scheme should be higher than the rate of the optimal scheme, right? Because it has a higher average rate. But of course, this is not true. What's happening is that our engineering perspective is making us too greedy. And what happens, because of that clogging function, I actually achieve a lower rate with the myopic scheme because I keep getting clogged. So the point that I want to make with this is simply that when we take these biological constraints into consideration, we often get results that may be non-intuitive for us if we had only looked at it from an engineering perspective. So I want to go back to this notion of communication over channels with state and remind you that that clogging function was a function of the state, and as a result, the problem that I looked at before was one of communication over channels with state. The difference that we now need to incorporate from the classical results, which only worried about communication, is that I need to take state estimation into consideration. And I want to highlight the fact that on Monday, we had an entire session on multi-access systems with state. And those are all very relevant to the kinds of problems that we care about here. So why do things change? Why is it different? Well, in contrast, what we typically do, where we worry about only decoding the message, now I also want to get state information at the receive side as well. And I might have differing amounts of channel state knowledge at my transmitter. We have a new function now, which we call the capacity distortion function, where capacity corresponds to how much I can communicate, and distortion corresponds to the fidelity at which I can estimate the state. Okay? And so here's a new function we can compute. Now, depending on how much information I have, things change. I want to just highlight some forms, if you will, of the result here. So if you look at the classical Shannon result for communication over compound channels with causal CSI, you see there too that there's a constraint on the input distribution. We get the same thing for the one-hop case where there's no channel state information, where we have to ensure that we optimize over input distributions that achieve that distortion at the receive side. If I now have some information, I get something that echoes what we see in the Gelfand-Pinsker result, which is I have to expend some cost in order to communicate the state to the receive side. And I also have my constraint over my input distribution. So this is nice, but of course, this is all one-hop results. So I'm excited about the fact that we now have two-hop results, and Wendy is going to be presenting those on Thursday, which means that I'm getting that much closer to being able to say something about my bacterial cable, because now I have a two-hop system where I want to estimate potentially both of the states at the receive side. You can further, so here's the results, again, echoing now everybody. We have Shannon, Gelfand-Pinsker, and our classical cut set bound here. But what I want to point out here is that we now have results where we can look at the optimal length of one of these relay systems. So this is a multi-hop system where we assume everything is Gaussian, everyone has the same distortion, and you can see along this axis I have the number of hops, and at some point the cost of conveying information becomes so burdensome that my rate drops, okay? So is something like this happening in one of these biological systems? I don't know. What we do need to consider are correlated states. This seems to be more legitimate for this variation of environmental conditions. And we also need to work more carefully to understand what are the relevant models for state that make sense for these biological systems. But I'm hopeful that maybe we'll have something to say to you next year at ISIT. All right, you've been very patient. So let's get to the more complicated network situation here. And here too, Nicola played a large role in the work that we've been doing on a problem called quorum sensing. And we were assisted by another biophysicist at USC, James Bodeker, and then a new postdoc, Marcos Vasconcelos. So quorum sensing 101. Just out of curiosity, how many of you know what quorum sensing is? And not because you just read the slide. Okay, Chris knows there's a handful. This is a very interesting and exciting phenomenon. We have our simple unicellular organisms, our bacteria. They generate compounds that are released into the environment. They have the capability of recognizing the presence of these molecules and compounds in the environment. And if there's enough, they actually fundamentally change their behavior. They express new genes. And so we have a, if you will, communication, sensing, and then action problem here, where everything changes. Now, why do we care? Well, we care for a variety of reasons. I'm going to emphasize this notion of bioluminescence. People had seen this species of squid, and it bioluminesced. At first, they thought it was the squid that was luminescing, and then they realized, no, it was the bacteria on the squid that was luminescing. It's what governs biofilm formulation, and if you want to build a microbial fuel cell, you worry about this. And for my particular colleague, James Bodeker, he's interested in this notion of virulence factors. So a lot of the infections that we get occur because the bacteria in our body quorum sense. All right? And so imagine a universe where we can potentially prevent infection without having to resort to antibiotics. So this is the holy grail of why people study these problems. So again, the key thing here, and the reason why it's called quorum sensing, is that the belief, well, it's been measured, that the amount of autoinducer in the environment is directly proportional to the number of bugs in the environment. Okay? So you only want to quorum sense when there's enough of you, because it's actually expensive to express this gene. One point that I'll highlight, and I should have mentioned this for both my biophysics colleagues, is that James' claim to fame is that he got a single bacterium to quorum sense because it essentially smelled itself. And it's hard to do. It's a microfluidic challenge. Imagine creating a device small enough that you don't have edge effects, but the bacterium can be fooled. And Mo, what his claim to fame is recognizing that these bacteria, certain strains, Shewanella onidensis MR1, affectionately known as Shui, forms nanocables of electrons between each other. And so I'm very privileged to be working with the hot young biophysicists in microbiology. Okay, but the granddaddy of them is another USC colleague, Ken Nielsen. And he's actually the one who first determined the mechanisms by which bacteria quorum sensed. And this is the key paper, and this is an image of the bacteria bioluminescing. So this is Vibrio fischeri. He's an amazing individual, has made profound contributions in microbial fuel cells, et cetera. Okay, so I should have emphasized, or maybe I did, but I'll remind you again, I'm not a biologist, I'm not a biophysicist, I don't even play one on television, but I've been talking to a lot of them, and so I've learned what I needed to learn to develop my models. So in order for these genes to express, certain key compounds, if you will, have to bind to important points on a gene. The thing that I want you to see here is that I have to have multiple bindings that occur in order for this gene to get expressed. It's not a simple system. Okay? People have studied in our community this process of binding. You can model things as a simple two-state Markov chain, and you can look at questions of capacity, et cetera, in order to understand these. When we started to talk to our colleagues, we realized that it was a little more complicated than that. So bear with me. So the compound that we care about is this autoinducer, AFT. It's my only global parameter that I'm going to have. I have receptors that bind to these autoinducers to form complexes, Those complexes, in turn, bind onto different sites on that promoter gene that you saw. And depending on where they bind, one of three things can happen. They can create the machines, synthases, that create more autoinducers. They can create new receptors, and then they can also, if you will, cause infection. So the red boxes are bad. All right? Each of these have unique binding rates. And I'd like to point to this invited tutorial that I asked James and Ken to write on quorum sensing in a new journal, which I'll talk about at the end. So here's my single bug, always a rounded rectangle. And you can see I have all of these different parameters that I need to keep track of, all right? The number of synthases, the number of receptors, the number of complexes, they have these binding sites. Now, if I'm talking about a quorum sensing environment, I'm not talking about 1 bug, 10 bugs, I'm talking about thousands of bugs. And I just told you that one of the things that I want to do is do computations, simulations, optimization. So you should have a minor panic attack right now that the number of parameters that I have to worry about is going to be too large. But again, we are engineers, we are theorists, and statistics come and help us out in this case. And that is, I can show that the totals are sufficient statistics. So I don't have to keep track of each of these individual numbers for every bug. I just have to worry about the aggregates, the total number of complexes, the total number of synthases. What can I do with this, right? This is one of my queuing systems again. I can look at certain values. What is the cell density? How many autoinducers do I have in the environment? And here too, we were able to take data from James's lab. And this is looking at the cell density as shown by, the experiment, which is the solid blue line. And our queuing model are the pluses here, all right? And once again, you see we get incredibly good fit. Of course, the dirty little secret is that this is for the closed system for E. coli. The way you measure what's going on in terms of quorum sensing is that these bacteria have been engineered that when they express a new gene, they light up. So you're doing a visual counting of what's going on. One thing you should know is that not all of the bacteria quorum sense, even though the bulk of them might. And this is something that we're interested in. So there's an excellent match here. I have 3 other curves that don't match as excellently. So there's still more that we have to do, which actually brings me to, wow, my next problem, which is that in the previous case, we were just measuring populations of things, right? We were trying to understand what was the cell density, what was the value of the autoinducers, and things like that. But the decision as to whether one should quorum sense or not was not included in our model. And so in this last piece of work, what we want to look at is actually thinking about these systems as if they are decision makers in uncertainty. And this is the new work that Marcos is doing, which I think is extremely exciting. So, I have to jump ahead a little bit more, sorry. So now, if I think about these as decision makers, I get into the realm of networked decision making. This is a very active topic of research in the controls and environment. And so I have some state of the world, I have a collection of observations, and each one of my decision makers can potentially take an action. But that action is going to have a cost. And so we want to understand, is it worthwhile for everyone to take this action? Who's going to take action? Can we say other things about this? Oh, I should note that in the end, we're going to model this as a game and look at game theoretic types of results. We are not the first to look at game theory for studying bacteria. Mihaela van der Schaar has a really nice paper where they look at the formation of biofilms and whether bacteria form connections or not, also modeled as a game. So we're going to assume that that global variable, the autoinducer, has some probability distribution. And so my observations at each of my nodes is going to be some kind of Poisson random variable as dictated by this X. But now, each of my bacteria have a strategy that they can employ. They have to make the decision. Is it worthwhile for me to quorum sense, given that this is an expensive thing to do? Even though I know if there's a lot of us, this will provide biological fitness and make the entire population happier. So you can set this up as sort of, a classic kind of strategy where you look at public benefit versus cost. And you can do some computations. And so one of the interesting things that we can now do is make a prediction as to how many of the bacteria actually are going to quorum sense. And now we have a situation where we can go back and look at James's data and see if we're modeling this uncertainty in the environment accurately enough. Now, of course, there are other things that are not present here. My model, on the one hand, by looking at decision-making under uncertainty, does not consider all of those complex processes that I told you about in terms of how autoinducers are constructed. And that's something that we have to work into the model. So, do these threshold strategies represent Nash equilibria? And I go back to my favorite question, is Mother Nature optimal in any way? I hope yes, but I want to paint the picture for the fact that there's even more work to be done. In my original modeling with Niccolò on the quorum sensing, we looked at a single signaling mechanism, one autoinducer. This is the signaling mechanism for Pseudomonas aeruginosa, which is the bacteria that my colleague James is interested in. That LASER protein, that's what we worried about over here. But there are 3 other mechanisms that we need to worry about. So we come to the point where we have to ask ourselves, how much of this complexity should we work into our models? At what point have we abstracted too much or too little? And I would maintain that we're not there yet. So what have we done? We've looked at biological systems through 3 lenses: point-to-point communication, multi-hop communication, and decision-making networks. I think we have some interesting results. It's certainly not the total story. We are very much happy about the fact that when we've looked at actual data, we've gotten very good match, but there's still a lot more to be done. So, I want to end by putting in a plug for the new journal. So the Communication Society started a new journal, the Transactions on Molecular, Biological, and Multiscale Communications. I did not create the title. And to be honest, Andrew Eckford was the one who did a lot of the heavy lifting in terms of putting this journal together. I feel that I'm merely the caretaker. What I'm showing you here are the covers of our 2-part special issue on information theory and biology. And so these came out, I think, in November and December of 2016, respectively. Many of you in here have taken part in putting papers together here. I want to encourage you to submit your work. A lot of the biological journals are very data-driven. They're not interested in optimization or theorems. I am. I'm the right home for those kinds of papers where I will allow you to be abstract. I will also allow you to devise models from data. But we're very excited about this, and many of you are taking part in the journal as well. So I will end there. Thank you very much for your attention.
Thank you very much. So we have time for some questions.
Andrea. Right. So let me translate your question. So Andrea's question was, in these biological environments, how do we even know what the right objective function or cost metric is? And this is legitimate, so I don't have a good answer. What I can say is that I believe we can reverse engineer things such that we can potentially make predictions about environmental conditions and say, given that we saw this experimental data and this was the result and this was the length of the cable, we can conjecture that maybe this cost function is an important one. And so it might have to be system design where you look at a variety of them and see which one works. But I think we're in the regime now where they don't understand at all why this is happening. And what's also interesting that I didn't say is that in this multi-hop cable, I have 2 highways. I have my electron superhighway, which is the sheath, but I also have an interbug highway, which is much slower. And so clearly these 2 highways exist, and we don't know why. I'm going to take the liberty, if you don't mind, because Andrea asked the first question, to tell you a little story about how I've actually been thinking about biological problems for a long time. And this actually, I like to call a tale of 2 fathers. Some of you may know my father is a relatively well-known professor of image processing and signal processing. And when I was an undergraduate, I was determined to be an electrical engineer, but not his kind. And I strove to figure out what would I do. And there was this new subdiscipline within EE at Berkeley, and it was called bioelectronics. So I took a few classes, and it seemed kind of okay, but it was mostly instrumentation. It was not what I wanted to do. I didn't want to build measurement devices. And then I took an intro to bioengineering class from your dad. And this is where, so I think it was 1987. I tried really hard to find the course catalog, but they had not, scanned it every single page. And so, the other Professor Goldsmith is the one who taught me about differential equations and how they related to diffusion. And I felt that the universe has kept pushing this problem on me, And so when I was assistant professor at one point, I was at an NSF meeting where we were, I think, once again, debating the death of coding theory and where should communications go. And I remember saying, well, what about biological communications? And this was probably, 1997 or something like that. And a senior colleague said, well, what does that mean? And I said, well, I don't know. But 20 years later, I think I do know. And then at my first sabbatical, I got some Princeton promotional material on Bounty Bassler, who's very well known for her work on quorum sensing. And at that time, we were all worried about sensor networks. And I thought, quorum sensing, this is the ultimate cooperative communication network. So every 4 or 5 years, the universe says, Ubli, you should think about this problem, and now I have finally listened.
Would there be another question?
Bless you, Sergio.
I thought I might just ask a quick question.
Sure.
So I think you maybe covered this to some extent, but it's so exciting to see all these new opportunities for our tools and techniques. But have you also, how much have you found that in the end we need new tools and techniques that we've got to develop rather than the tools that we already have?
So I don't know if you were looking at my slide, but if I can take the liberty of skipping to the next one. I think our tools and techniques actually still have a lot to say on small problems. So one of the things that James and I have been talking about is multi-species interaction for quorum sensing. And what's interesting about here is that different species of bacteria typically coexist, and they create different compounds. And these different compounds can do different things even to our simple one-hop channel, which is that one compound can bind to the site of another, preventing bacteria A from quorum sensing. It can modify the binding site. It can bind, and the bacteria can think that more of its family members are there when in fact they're not. And so I would maintain that even with the tools that we have, like descriptions of erasure channels and jamming, we still might be able to say something interesting. So we're so far away, I am so far away from solving some real problems that I think these baby steps are necessary in order for us to move forward. And one thing I will say, at least for my colleagues, is that they are interested in operational behavior. They don't want to simply emulate what they see in the data. They want to try to understand what's really going on. And I think in that regard, we have a common philosophy, if you will, in terms of operational behavior from an information theory side versus simply emulating behavior.
I think you'll all agree with me that was a fantastic keynote. Let's join together and thank Ubli again for a wonderful talk.